right, so you need a transport protein for facilitated diffusion, just like you do for active transport. The difference is that with active transport, where are you going?
Against the concentration.
Against the concentration gradient, so therefore it takes energy. It's like you're going uphill. You should know that the prime example of active transport is the sodium-potassium pump. I've even seen essay questions where you got a point from knowing that it's 3 sodiums that are going out for every 2 potassiums that are coming in. Is that right?
Yeah, there is.
Potassium is released. So potassium, this is the inside of the cell. Potassiums are coming in, sodiums are going out. So 3 sodiums out for every 2 potassiums coming in. Got it? So this is simple diffusion, high to low, without the use of a membrane protein. Facilitated diffusion, high to low, but with the use of a membrane protein. Neither of these require energy, so they're considered passive. Okay? And then active transport against the concentration gradient uses a membrane protein, has to have ATP.
And it doesn't have phosphorylate the transfer protein.
Yeah, to change its shape. Okay, and then exocytosis is bulk transfer. And then, I don't have a picture of that. That's unfortunate. Exocytosis is when you're going to have a vesicle to dump out like a lot of something, but since you're having to rearrange the plasma membrane, it is going to take energy. Okay? Endocytosis, same thing. It's going to take energy. You're pulling in bulk stuff into a vesicle, and it'll go wherever. If it's solid material, then we call that phagocytosis. If it's liquid material, then we call it pinocytosis.
What
Right?
things would people try to be?
Huh?
What type of things would they try to do?
Food. If it's a macrophage, then it's consuming pathogens. It could be consuming cell debris, like red blood cells that are digested. You might need to, or if you have damaged cells.
Yeah, it'll consume it and then break it down and use the stuff to release stuff.
Good. Receptor-mediated endocytosis means kind of like if you're going out to a club, it's not good enough just to go through the door, but you've got someone checking names. Is your name on the list? Well, then you can't come in if your name's not on the list. So, receptor-mediated endocytosis. So that means there is a receptor at the plasma membrane, and only things that match with the receptor are going to be able to come in. It will not open. Like, remember we did ion, like ligand-gated ion channels? You have to have that ligand in order to open up the gates for those ions to get out. Without the ligand, then the gates stay closed and the ions don't get to move through. So there's a receptor involved that mediates whether or not those channels will open or not. Good? Alright, so that's it for chapter 7. Okay, so let's stop there. That's a good place. And we'll do our 8